everyone, so today we have the second part of DNA Forensic Fingerprinting Lab, and we start with the preparation of 1x TAE buffer. So the stock one is 50x, and you need to prepare 2 liters of 1x TAE buffer. So here in your lab book you will need to calculate and document the volumes. Then you will be using this 1x TAE buffer for the gel preparation. So you will be casting gels with the agarose dissolved in this TAE buffer. And also it is going to be used as a run-in buffer for gel electrophoresis. Okay, so the next what you do, you cast the gels. First of all, of course, you weigh 1 gram of agar or agarose, and it is going to give you 1% gel. So you weigh it, then you put it in the Erlenmeyer flask and bring to volume of 100 milliliters. Of course, you dissolve it in TAE buffer. But the thing is, the agarose is not easy to dissolve, that's why you're going to heat it. And heating is going to be performed in the microwave. You could also use the heaters, but it takes much more time. So for the microwave, you're going to use the maximum power, and the time is 1 minute 30 seconds. So in this case, every 30 seconds you're going to check your gels, and you don't want to boil it, you just want to dissolve it, okay? So every 30 seconds stop the microwave and check the solution. When it's totally transparent, you can remove it from the microwave and let it cool down on the bench. While it's cooling down, you can assemble the forms in which you are going to cast the gel. Then, after we obtain about 45 degrees of Celsius, we can slowly pour the gel or the solution of the gel into the form. And you want to avoid the formation of bubbles, because if you take the gel, so let's say we have 3 wells here, and at some place you will have a bubble. When your DNA is applied the current, so minus and plus here, it will move towards the plus pole, and if it encounters the bubble, it will kind of try to go around it, and of course in this case you will not receive the clear result. So let's say that we have 2 parts, one was going straight, another one was going through this bubble, and in the result you will obtain something like that instead of a good band like this. Okay, so you leave it at room temperature to solidify. In the video you will see 2 types of gels that we're going to use. One type is usual, and we're going to stain it with fast blast dye. The second one is actually fluorescent, and fluorescence is given by the Cybergreen fluorophore, Cybergreen. So what happens here, the Cybergreen is positively charged, and in this case it will move from plus to minus, right? And whenever it encounters the DNA, it will form a distinct band which will be visible under the excitation with UV light. So we will be using UV light, okay? So the next video would be the preparation of your samples for the real gel electrophoresis run. And first of all, you are given the loading dye and the standard DNA. So it is the digest of Hint3 lambda enzyme. So this will give you the different samples of DNA. So it will look like a ladder, but of course it's not a ladder, it's just different sizes of the DNA cut by this lambda digest. Okay, so the 5x loading dye, you will need to add it to your samples, and your samples are 20 microliters. Hint3 should be also 20 microliters. So you will be calculating this volume of loading dye. Of course, you should document it and write it in your lab book. Okay, so here we have everything. So after addition of loading dye, you should spin down these samples using microfuge so that in your tube they really could mix. So let's say that this is your sample and this is your loading dye. If you don't bring them down, you won't be able to mix them properly. So you spin them, everything goes down, and in this case, of course, we have the mixture of your sample and loading dye. So in the fourth video, you will see how to load the gels with the samples, and first of all, you're going to pour 1x TAE run-in buffer into the gel tray. So usually it takes about 1 liter, and this run-in buffer should cover your gel and also fill the wells in your gel. Okay, so what you do when you load it, first of all, you take the sample and everything is spun down. Next, you're going to place the tip of your pipette inside this solution and pipette it up and down so that you mix it well again. Okay, so after that, you're going to take 20 to 25 microliters and place them in the well. So let's say this is a well of your gel. Okay, so here should be the level of your run-in buffer, and you should place a tip of the pipette inside the well. If you are putting it above it, the part of it will just float away. So don't do that. Try to really dip it in the well and then slowly release it. You push on the button on your pipette, which creates the pressure, so the samples will be released inside the well. Okay, do not release this button before you remove the whole pipette tip from the well, otherwise you will suck up the sample back, and also you will create the vortex here, which will lead to the floating of your samples again. Also, why you would need to do it slowly, because if you make it with a huge pressure, the sample will counterheat the bottom of the well and float away. Okay, another word for the loading dye. So why do we use the loading dye? In the video I said that we really need to bind the DNA to glycerol, and of course if we don't use the loading dye, the DNA will easily float away here. Okay, so be careful and use all of these tips. Of course, you can use your second hand as a block so that this tip will not be shaking, and of course it creates more precise conditions for your experiment. After you loaded all the wells, you will need to close the lid, and of course you have this tray here with 2 electrodes. One will be red, the second will be black. Black is usually for negative and red for positive. You should connect them color to color, so red to red and black to black. On the video you will see the four columns of the connections or connectors, and you will need to use these columns, okay, because they are related to the same output. If you plugged red to this connector, you should plug the black to the same column connector of the black. So this is the right way to connect. If you want to use red here and black here, it won't work, and you will see the error message on your screen. Of course, the wells of your gels should be closer to the black pole. Why? Because the DNA is negatively charged and it will move towards the positive electrode. If you connect black to red and red to black, you will have positive here and negative below. In this case, from your well, so let's say this is your gel, and here is the loaded well. Of course, the DNA will easily go through this part and you will lose the experiment, so be careful with the connections. Black, which is negative, is on the top of the gel tray, and you connect it to the negative connector, okay? So after you connected everything, you should set up the power supply, and we're using 120 volts. It is the constant voltage. Also, you could increase it to 130, but not more usually. You could also decrease it to 100, but in this case, of course, it takes much more time than 120 voltage. After that, you're going to wait until your bands travel on your gel about 2/3 of its length. If we wait more, we are in risk of losing some smaller DNA fragments. So usually 2/3 or even half of the length of the gel. In the next video you will see how to dilute the Fast Blast, and we are going to use 5X working solution. So for this staining I am preparing 500 milliliters, and it is our final volume. This is our final concentration, and the stock concentration is 500X. Now you need to calculate how much of Fast Blast stock you need to dissolve in how much of water. Of course, you document it in your logbook. So you collect the gels from the tray and rinse them in distilled water. And after that, you can pour the Fast Blast working solution, and you should cover your gels with the Fast Blast dye. Otherwise, they will easily dry out and you will not be able to take a picture of these gels. You usually put it on the agitator or the shaker, it is also called shaker, and leave it overnight. After you put it on this shaker, you should set the appropriate speed, because if it's too much, the Fast Blast will be spilled from your gel holders and they will dry out, so be careful with the speed of the shaking. Okay, so the results will be looking like this. So this is the Fast Blast dye, and this is Cyber Green. Cyber Green is the fluorescent dye, which could be easily imaged after excitation with the UV light. So you apply UV here and they start glowing. Okay, so you have Hint3 DNA standard, then you have crime scene, suspects 1, 2, 3 and 4. In your results you should write how much the fragments traveled from the wells, and you could do that by knowing the distance from this point to this point. So usually our gels, they are 8 centimeters. So if you know these widths here, you can easily calculate the distance from here to here and here. Okay, so this is also left for you to calculate. So calculate the distance after you know the width of the gel. So it is again 8 centimeters. Here you can see the image of the Fast Blast stained gel. So what is going on here? So the Fast Blast is positively charged dye, and it goes through the gel to the negatively charged DNA. It binds to it, and that's why we can easily see the bands. So sometimes, because it also colors the gel, we don't see the smallest or faintest bands, but if you compare them to the Cyber Green gel, you could see that here actually we have one band, as well as here and here. So sometimes it's easier to do the Fast Blast. Of course, if you don't have Cyber Green, you could use ethidium bromide. But ethidium bromide is toxic, so you should be very careful by using it. And Fast Blast is generally accepted to be used in the educational labs. Okay, so I will post these pictures and you will be able to calculate them, the distances, even in the image editors. So there is a special tool called ruler, and with ruler you can easily calculate the pixels, and while knowing the standard width, which is 8 centimeters, you could easily calculate the distances in the image editors. Okay, thank you all for listening. Goodbye.